session. Thank you for joining. My name is Gerben Molhuis and I'm with ISA Pharmaceuticals in the Netherlands, and I'll talk about our company. I think it's fair to say we're a true pioneer in targeted immuno-activation, finding ways to activate the immune system to treat chronic infections and cancer. Built on a very strong, best-in-class platform, generating specific T-cells, leading science, but meanwhile progressed to be a late-stage company, in essence only one trial away from product approval, in partnership with our US partner Regeneron Pharmaceuticals, a $40 billion Nasdaq-listed, high-ranking US partner. And we're here raising funds for our next products in our pipeline. With our lead product being late stage, we focused on building up a portfolio in orphan diseases. In a nutshell, our technology is actually very elegant, very simple. It's about injecting long peptides, just a traditional sub-Q injection or intradermal injection, and then let the immune system do the rest. Actually, long peptides are taken up by cells of the immune system, are processed, and then one of the hallmarks of our technology is that we generate both CD4 and CD8 positive T-cells. CD8, I think you all know as the killer cells, but the CD4 helper cells are probably equally important for memory, but also for direct tumor effect. I guess in this audience, everyone is aware that the T-cells are the workhorses of immuno-oncology. The T-cells are the cells that actually recognize the tumor or the infected cell and kill the infected cells. I think you're probably also all aware that cancers by themselves are not very immunogenic, typically try to do everything to escape a tumor response. But also checkpoint blockers, the 10 billion class of drugs that came in being only a few years ago, in itself does not generate specific T-cells. All quote-unquote checkpoint blockade does is take away the brakes from the immune system, but you still need the T-cells to do their job, and that's where our technology comes in. We believe by adding long peptides to standard of care cancer treatment, you can really improve efficacy, and I'll have some clinical data to support that. One added advantage of the technology is that it is very safe. It does not add a lot of systemic toxicity. It's a little bit of local site injection reactions, but in terms of systemic toxicity, the technology is proven to be very safe in over 150 patients. And that makes it, in our view, an excellent choice to combine with other modalities. And our view of the future of cancer is that the SLP technology is there to play a role together with the well-established players, obviously anti-PD-1, that enables T-cell infiltration, T-cell functioning, improve the tumor environment, but also in our view chemotherapy has a role to play, has its own direct cancer cell killing, but also an effect on the immune system. And I'll show you a little bit of data of all the work we did on the role of chemo. So the three modalities, SLP, anti-PD-1 or checkpoint blockade, chemotherapy, in our views are very synergistic and can form the future of cancer care. We demonstrated combination between SLPs and chemo. We also demonstrated in the clinic synergy between SLP and anti-PD-1. I'll take you through some of the key data. And we were very encouraged by the data presented last year in lung cancer, combining anti-PD-1 and chemo, actually very much supporting our views on synergy between anti-PD-1 and chemotherapy. In terms of our product pipeline, we focused historically a lot on papillomavirus-induced cancers, HPV-induced cancers. Our lead product targets a specific papillomavirus type, HPV-16, and is now in 2 late-stage trials together with anti-PD-1, one in head and neck cancer and one in cervical cancer. We meanwhile identified 2 next products in our pipeline, ISA102 and 103, targeting the non-HPV-16 cancers and targeting PRAME, another tumor-specific antigen. I'll cover that in a minute. And then we have a platform that's able to do much more. It can generate other SLP mixtures targeting other targets. But right now, in terms of fundraising, our focus is to bring our next 2 products to clinical proof of concept in parallel with randomized trials for our lead product, ISA101. Well, as I mentioned, we've been very much focused on establishing and understanding our technology, generated a lot of data in multiple trials in over 150 patients. We did some early work on the combination of chemotherapy and long peptides and actually trying to understand the immunomodulatory role of chemo, so the effect chemotherapy has on the immune system. And very nice paper already in 2016 describing this work is in Science Translational Medicine, and just in a nutshell we showed that with the standard of care chemo in cervical cancer you actually can reduce the myeloid-derived suppressor cells, immunosuppressive cells in the blood, but you actually do not affect the lymphocyte population, the population that's responsible for generating T-cells. And after 2 cycles of chemo, it's a little bit difficult to see here, but you can see these suppressor cell populations are back below where they historically or traditionally are in healthy people, and the lymphocyte population is unaffected. And if you use that by coming in with your vaccine, your SLPs, after the 2nd cycle of chemo, you're actually able to generate very strong tumor-specific T-cell responses. And that's what we explored in this trial, about 80 patients. This was a dose-finding trial, but also the timing of the SLP technology combined with chemotherapy was investigated here. And we also showed in this trial a very strong effect on clinical efficacy, a strong correlation between the HPV-specific response and survival. Too much detail to go into for this presentation, but happy to answer questions if you're interested. And we also showed that actually that survival benefit was really driven by the tumor-specific response and not by the general health or immune status of the patient. So that's combination with chemo and long peptides. Another set of key clinical data follows from the work done at MD Anderson, where we combined the long peptides with anti-PD-1. This is nivolumab in this particular case. You can see the trial design here. This was a single-arm trial, optimal dose selected from previous trials, combined with nivo in mostly chemo-resistant head and neck cancer patients. And we know from now, data that was published in parallel with us generating our own data, we know that nivo on its own, so anti-PD-1 on its own in this population, has about a 16% response rate. So about 16% of the tumors shrink, and median overall survival of the monotherapy of nivo is about 9 months. Well, we showed that if you combine our long peptides with anti-PD-1 in this setting, you actually more than double the response rate. These are the data from the trial. About 36% response rate in the head and neck cancer patients. Very interestingly, we see 2 complete responders, and we now know that they are very durable long-term responders, very different from nivo monotherapy. We also showed a doubling of the median overall survival in this trial compared to nivo monotherapy. Well, again, very encouraging data combining anti-PD-1 with our long peptides, very consistent with the scientific data we had generated all along on how this technology works. Well, it led us to partner this product with Regeneron, and together with them we're now running 2 clinical trials, and as I mentioned earlier, we're in essence 1 trial away from approval for this product. These 2 trials should lead to approval of ISA101. The first trial is in HPV-16 positive head and neck cancer. It's a 2-arm randomized multi-center controlled trial, 164 patients, 50 centers, 10 countries. Where this is a trial run by ISA, and actually we announced just earlier this year we dosed the first patient in this trial, and we have most of the approvals in, IND accepted, most of the European approvals in, and we expect to have readout from this trial by the second half of next year. The second trial we'll do is in cervical cancer. This will be a randomized trial again in phase 3 trial in first-line cervical cancer. This trial be run by Regeneron. And very interesting for us, this is a trial where we will test triple therapy. So these patients will get standard of care chemo, plus anti-PD-1, plus SLPs. And we, based on all we know about how this technology works and the synergies we've seen, we're very encouraged, and we look really very much forward to getting the data out of this trial as well. Well, ISA101, as I mentioned, readout about 2 years from now in the first indication from randomized trial. And in parallel, our goal is to bring 2 more products to clinical proof of concept. The first one is a very obvious one. HPV-16 is responsible for about half of all the HPV-induced cancers in cervical cancer. So there's another big group of patients left that we cannot treat with ISA101. So we identified another product opportunity we call HPV-X, aimed at targeting the other half of patients, but very much following the same pathways as we did for ISA101. So we'll take the exact same treatment paradigm, treatment algorithm, just replace ISA101 with products targeting these other HPV types. It's about a $500 million market opportunity, and we believe we can get to approval very efficiently, leveraging the data we'll get out of this large phase 3 cervical cancer trial. And interestingly, all rights regarding HPV-X are still fully retained by ISA. This is outside our partnership with Regeneron. So we have an orphan drug opportunity that is very significant and requires limited investments to get to approval in about the same timelines as ISA101. And the second product we identified is a product called PRAME. That's a tumor-specific antigen. Originally discovered in melanoma, but actually overexpressed in a large number of cancers. We like it a lot because just like HPV, this target is functionally important for the malignant behavior, and overexpression of PRAME is clearly associated with poor prognosis. It's an intracellular target, so it's not accessible by antibodies. We do see the first T-cell type approaches getting to the clinic targeting PRAME, but we believe with our long peptides we have a much more efficient and a much more straightforward approach of generating T-cells targeting PRAME. We identified and designed and patented our long peptides targeting this target, and we're now preparing for clinical trials. We see a bit of a mixed approach here. There are a number of orphan indications where PRAME is overexpressed, like in sarcomas or in AML, hematological cancers, but also some of the more mainstream indications. For example, triple-negative breast cancer is about 95% PRAME positive, but also certain cases of lung cancer is high PRAME overexpression. And again, we see a role there of adding SLPs to standard of care, just like we've shown for head and neck cancer or for cervical cancer. So in a nutshell, I think exciting times ahead. In the next 2.5 years at ISA, we'll generate randomized data with our lead product, partnership with Regeneron, triggering hopefully substantial license revenues under the deal we have with them, and bringing our product to approval. It would be a major accomplishment, obviously. But in parallel, we expect we'll be able to generate proof of concept for our next 2 products in 2 different indications, targeting orphan diseases with potential fast path to market, and a technology platform that's capable of doing much more. There's a whole list of other targets we would like to work on. We have some early work on hepatitis B, for example. We've our toes into the neoantigen space, so there's much more to follow from the platform and from the technology. And with that, open for questions.